Hi, my name is Madeline Schmitz, and I'll be presenting the CRAS protein today to my grandparents who were both English teachers. In your body, the CRAS protein is mostly responsible for cell proliferation, and it's most commonly found in your epithelial cells, which are the outer lining of a lot of your organs, and on your skin, your lung cells, and your colon cells. The CRAS protein is not present in plant cells, and the cell is the basic unit of life for the body, and there are billions of them within your body. They make up all of your organs and are how we are alive and functioning today. But the CRAS protein, since it's so responsible for cell proliferation, is connected to cancers very closely. Most commonly lung cancer and colon cancer. So there are two types of lung cancer that are the most prevalent cases of the KRAS G12C mutation, which I will get into in a second, and that's small lung cancer and non-small lung cancer. And the difference between these two is the rate at which tumors spread within the lungs. But those two together comprise about 40 to 50% of all patients with this KRAS mutation. So this is a model of a normal KRAS protein, which are found on the inner membrane of your cells in your body. And these Q-tips, these green Q-tips, are the glycine amino acid in the sequence located at codon 12, which is stop 12. And amino acids are another structure of life which comprise a lot of our protein. So this is a really small unit of a protein. And glycine is located more towards the outer structure of the cell, so not in the immediate center, but also not on the outside. And for my structure, I made 3 little dots on each head of a Q-tip, representing 1 amino acid, 2 amino acid, and then put together to form an amino acid chain. So that's the primary and secondary structure of this protein. The tertiary structure is all of these amino acid chains put together to form a KRAS monomer, which is single. And that's a singular form of a lot of protein. So this is a monomer of KRAS. Put together with another regular KRAS protein, you would have a polymer, which would be the quaternary structure, which means more than one of a protein put together. Quaternary structure is the final structure of building for proteins. So here is my mutated KRAS protein. And in the G12C mutation of KRAS, cysteine replaced glycine at codon 12 of the protein. And I might think, why? Like, what's going on? Well, when cysteine replaces glycine, cysteine leaves an open pocket in the protein because it's not supposed to bind there. And so that's why this dice is here to represent cysteine, because it's a little bit more flat and it's a druggable pocket for other proteins to bind in the KRAS protein. So when cysteine is here, other proteins are able to come in and bind on the KRAS, making the KRAS protein always in an activated GTP state, which GTP is energy. So if the KRAS protein is always producing energy, it's always active, and the KRAS protein is driving cell continuous proliferation. So that's how the protein function and the cell function works with this mutation. For the organism is eventually going to develop a cancer, either in the lungs or the colon most commonly, since this all activated cell proliferation often results in tumors. And like I said, both of these, glycine and cysteine, are located at codon 12 of the KRAS protein. It's just which one binds to the KRAS protein in order to make up this chemical makeup. 3 limitations I found for my model are that my regular model is not as circular as the real KRAS protein within our body. I did my best to make it appear 2D circular, but 3D circular is a little bit difficult unless I had cotton balls. And quite obviously, there are 188 to 189 amino acids present in a normal unmutated KRAS protein. And within my model, there are probably about 60. So I have a third of what the normal cross protein would have. And my model is, it's very hard to depict really tiny amino acids, so the Q-tips might be a little bit of a too big representation of glycine within the entire protein, but I wanted to make it so that the audience could see it compared to the dice of cysteine. So yes, thank you so much for watching. Have a great day.